welcome, good evening, and welcome to the second session of Out of This World Healthcare, Space Medicine and its Influence on Earth. It's my pleasure tonight to introduce you to a distinguished member of the UC Space Health team, Dr. Tammy Chang, who is an associate professor of surgery in the Division of General Surgery. Before I get into her intro, I also want to introduce my co-chair, Dr. Stephen Robinson, who's joining us from hundreds of miles off the surface of the Earth or another planet known as UC Davis. And we're really happy to bring to you Dr. Tammy Chang tonight. She is a gastrointestinal and acute care surgeon in the Division of General Surgery here at UCSF. Her clinical training includes internship, residency, and chief residency in surgery at UCSF. And she did her undergraduate work at UCLA. So a real UC alum and fan, and we're really happy to have her in the Space Center. In addition, she completed a clinical fellowship in minimally invasive surgery and bariatric surgery at UCSF as well. Her research experience includes a postdoctoral fellowship investigating three-dimensional organization of hepatocytes in rotational bioreactors. And part of her inspiration is in the UC system, but I have to give a little credit to Harvard as well, where she did her MD, PhD. Her background demonstrates her strong commitment to facilitate transfer of advances in laboratory research into clinical applications that benefit patients. She holds her MD, PhD, as I mentioned, combined degree from Harvard Medical School through the Medical Scientist Training Program. And her PhD is in the field of immunology with a specific focus on immunoregulation in autoimmune disease. And this is going to be an exciting talk tonight where she explains to us a field that is incredibly complicated, but with her visuals, I think we're all going to come away with a much better understanding of the long-term goals of tissue engineering and basically developing a basic liver unit for therapeutic implantation and how she leverages the space environment to accelerate that work. I also want to remind everyone that we've dedicated this series to our dear friend, mentor, colleague, inspiration, Dr. Millie Hughes-Fulford. And I'm very pleased that Tammy will be giving a deeper introduction of Millie tonight. So Dr. Chang, thank you so much for joining us, and please take it away.
Thank you so much, Ainur, for that wonderful introduction. So I want to begin with talking about Millie, and she passed away last month and was a great loss. And I want to share some personal stories with Millie, and I think her accomplishments are many. To me, she was a friend and a mentor. And it's appropriate tonight because the reason I can give a talk on tissue engineering in space is because of Millie. She, as Ainur introduced, was my postdoctoral mentor. I did 2 years of research with Millie when I was a surgical resident at UCSF. And how I found her was I was interested, I started reading some papers about cellular aggregation, so cells coming together in microgravity simulated bioreactors that were NASA designed. And that really captured my imagination. Now I think there's a lot more work in these cellular aggregates, and you may have heard of them being called organoids in the press. And so there's a lot of things we're learning about organoids. But when cells are cultured in microgravity or simulated microgravity, they could form these aggregates, these organoids. And I was really fascinated by that, and I wanted to do research on it. And I went to the library, and I read some papers, and I found that Millie Hughes-Fulford was at UCSF. And that she was not doing research in tissue engineering with the liver, which is what I was interested in, but she was using microgravity to study the effects on bone and immune system. So I called Millie and says, hey, I'm a resident at UCSF. I read your papers. I'm interested in this aspect of microgravity and how it might be used for liver tissue engineering. Can I come work in your lab? And she said yes. And that's how it started. And I tell that story to illustrate what an open-minded, generous spirit Millie was. I mean, this was not, I came to her as essentially a stranger. It was not a scientific question that she was investigating at the time. But she welcomed it, she supported me, and that's how I got started in this field. And it's from the work that I did with Millie for those 2 years while I was a resident, that was the basis of my career today. And I will actually share some of that data and later on in the talk of how we got started. But very appropriate for me to dedicate this talk to Millie because she was how I got here. In addition to talk about tissue engineering space, which is what the main talk is about, I wanted to tell some stories about Millie because during the 2 years that I was working with her, I also had an opportunity to participate in spaceflight experiments. And we had some science adventures together. And one of them is going to the Baikonur Cosmodrome in Kazakhstan to launch spaceflight experiments. You may remember that the Columbia disaster, where the space shuttle was destroyed upon reentry and the crew was lost. And after that time, the science research in space halted, was stopped. And there was one particular project on the Columbia that the data was lost. And that experiment was called Lucan, where the goal was to study the effect of T-cell activation. And Millie strongly felt that to honor the crew that lost their life doing science, that we needed to continue this project. And she found ways then to keep doing the science. And that included collaborating with scientists in the European Union, and which at that time launched experiments from the Russian Space Agency in Baikonur. And we continued this experiment and took the experiment to the Baikonur Cosmodrome, which, as you can see, is a desert. And this is the Soyuz rocket that will launch the experiment, and it's carried on a train. And the Soyuz will then be lifted into the upright position. And this is a photo of the launch of the Soyuz rocket carrying the experiment. But this experiment came back with data, and it showed that there was very important differences with T-cell activation. The T-cells are a very important cell type for the immune system, and that these activation pathways were inhibited, and that this was a major way where astronauts may be more susceptible to infections in spaceflight. Another science adventure we had was to the Kennedy Space Center, and this was to launch another immunology experiment, this time with mice, looking at how the immune response is changed in spaceflight in an organism. And again, we had to pack up our lab, and we had to, and this is the Space Life Sciences lab on site at the Kennedy Space Center, where we set up shop for 2 weeks to launch the experiment. And really had an incredible time, visiting the site and being so close to the shuttle. This was SDS-131, so Discovery's second to the last mission. This immunology experiment was on board. And it was a morning dawn launch, and the little fleck of light in the distance there is the launch site. And we were 3 miles away watching the launch, and it was one of the most memorable experiences in my life to be able to watch a shuttle launch and then wait for our experiments to come back. And this experiment found that the spaceflight immune dysregulation was really, it goes both ways. And so sometimes when the immune system is too strong, where the stimulation is too strong, the immune system is supposed to shut down. But what we found was that there's the activation of T cells, these important cell types, are so affected that when they're supposed to shut down, they don't shut down. And so the dangers to the astronauts are in long-term space travel is immune dysregulation. So the immune system may not activate when it's supposed to, and may not shut down when it's supposed to. And because of the work of Millie and others, the immune dysregulation has really been recognized as one of the major issues to tackle in long-term spaceflight missions to moons and Mars as a health risk that we really need to address. And I think Millie's contribution to immune research and our understanding of the effect of spaceflight and microgravity to immune regulation is really a lasting legacy. So what I hope to cover during this talk is why tissue engineering is important, why we're even doing it, and what is the current state of the art of tissue engineering? What are people thinking about and doing and trying? And then hopefully convince you that tissue engineering in space makes sense and that it's an approach that is worth thinking about. So first of all, why tissue engineering? And this is a graph that motivates my work. And it's essentially what is going on with transplantation of organs in this country and worldwide. So for the last 20 years or so, these lines kind of continued the same trends coming out to 2020. You can see the green line is the number of transplants performed, and it's pretty flat. It's about more than 20,000. And the orange line is the patients waiting for a transplant at the year's end. It's a line, it's slightly increasing during the years, but you can see the large gap between the number of transplants performed and the patients that are waiting at the end of the year. And this gap is not because surgeons and physicians don't want to transplant more patients. This gap exists because there are not enough donor organs for all the patients that need them. And when I look at this graph, what it says to me is that there needs to be another approach. That transplantation is important, it changes lives, it saves lives, but there has to be a different approach in order to bridge these two lines. And to me, that approach is regenerative medicine and tissue engineering. When I was a medical student, I went to a talk by Dr. Joseph Murray, who won the 1990 Nobel Prize in Medicine for performing the first successful human kidney transplant. And he was a plastic surgeon by training. He really laid out the idea of the evolution of surgery in that talk, of the things that surgeons can do to help heal and repair. And first is to resect and repair diseased organs. And then after we do that, we can reconstruct it, we can rebuild it. And these are things that surgeons still do and do all the time to treat disease. And then in the next step, and perhaps because he pioneered this, is transplantation, taking organs from one individual to another to heal. But even back then, before regenerative medicine was really a term, he predicted or thought that the next step in the evolution of surgery is promoting or stimulating the body to regenerate its own tissues and using the surgeon's skills to help do that. And as a medical student, that really captured my imagination, but I didn't really know how that was going to look, how that would really be realized. But now I think there's several pathways to that, and tissue engineering is one of them. Well, I will be talking to you about liver tissue engineering, specifically for the liver, and that's what I work on. And I want to convince you that this is an important problem, a difficult problem, but a problem that's worth devoting effort to. And the reason is, well, liver is a vital organ, which means you need a functioning liver to live. And it's also the only vital organ that we currently don't have a human-made device that we can support survival for at least a little bit of time. If your heart fails, there's something called the left ventricular assist device that can help your heart pump blood for a little while. If your lungs fail, there's something called the extracorporeal membrane oxygenation, or ECMO, that can help you oxygenate for a while. And if your kidneys fail, there's hemodialysis, and a patient can be maintained on dialysis for years. But if one's liver stops working, we basically need a transplant. We don't have anything right now that can even tie you over for a little bit. There's some things on the market that maybe can give you a few days, but that's really a bridge to transplant, and it doesn't work so well, and that's why it's not widely used. And so why is that? Why are we haven't engineered something that can replace some of the liver's function? Well, when you think about the heart, if we want to reduce it to what the heart is really doing, the heart is essentially a pump, right? And so you can engineer a pump, a fancy pump, but it's a pump. The lung is actually really a oxygen exchange membrane, gas exchange membrane, and that what an ECMO is. That we can also do that. And a kidney is essentially fundamentally a filter, and that is engineered too. But what the liver does is hundreds of biochemical reactions, all regulated, responding to various inputs and outputs. And it's hard to machine that. And I would argue that to replace liver function, we need the hepatocyte, the liver cell, to be part of the solution. And understanding that is, and to make that happen is to understand how to keep the liver cell or the hepatocyte functional in a tissue engineered system. So these are all the things that the liver does. It regulates whole body metabolism. It regulates toxins. It clears bilirubin, makes bile. It makes proteins, important proteins in your blood. And the importance of the liver is illustrated by when the liver doesn't work. Almost everything goes wrong. Patients become confused, have encephalopathy. the fluid balance in a person's body is changed. You don't make the proper clotting factors. Patients don't excrete bile properly, and so the skin turns yellow, the eyes turn yellow, and the immune system doesn't work properly. And the liver also sits in a very particular place in the body, and anatomically it's in a very interesting spot because it has two main inputs of blood supply. One is the red, the oxygenated blood that most other organs have too, that comes from the heart. But the purple, or the portal vein, is blood that comes straight from the intestines. So everything we eat gets funneled and the liver sees first. And in fact, the portal vein, the blood that comes from the intestine, supports more of the liver's circulation than the oxygenated blood. And then the output is from the hepatic veins that goes back to the heart. So it sits in a very special place in the body with special inputs that is critical to its function. It also has a very complex microanatomy. It's pieced together in these little lobules, or these hexagons called lobules. The inputs, the portal vein, and when you sort of cone down and look at it, the inputs, the portal vein and the hepatic artery kind of goes in and flows toward the hepatic vein over there. And then there's a countercurrent flow of the bile that flows back to the bile duct. And all of this is highly vascularized. There's single cell or two cell thick cell layer thick sheets where there's blood flow, so that all the cells can sample and really act as a sieve and detect toxins that are coming through the intestinal circulation and do the metabolic regulation that it's intended to do. So when you look at this, you think, well, how are we going to make that? But when considering that problem, then you think, well, if we're going to have cells do it, what kind of cells can we use? Well, the amazing thing about the liver is it's a highly regenerative organ. So if we do a hepatectomy, or if we resect part of a liver, a healthy liver, that liver can grow back to its proper size and carry on function. And so in a healthy liver, the hepatocyte, the liver cell itself, is the one that regenerates itself. But in thinking about tissue engineering, and if we want to make a liver outside of the body that we could use to treat people, what cell types can we use? And this is where the thought of stem cells would come in. And in particular, the induced pluripotent stem cells, which are stem cells that are made from adult differentiated cells, is my favorite. These are cells that you can take a biopsy or a little bit of someone's skin cells or blood cells and put these transcription factors in and change them so that they could become a stem cell and become than any other type of cell in the body, including a liver cell. And one of the advantages of thinking about using iPS cells, or these induced pluripotent stem cells, is, well, for example, if my liver fails, then potentially I can get a little skin cell from myself, have it become a stem cell, and then differentiate into a bunch of liver cells, and then take those cells back. So if I'm taking my own cells back, then there's no immune barrier that has to be crossed in the case of current transplants. And so if you receive your own cells back, there won't be an immune response against that. So that's an advantage, right? But that's about sort of a custom making yourself something to wear. Which may work for some people. But what most people want is to be able to go to a department store and pick out something off the shelf that they could wear right away. And for a patient who is coming in with a vital organ not working, sometimes people don't have time to wait for this process of growing your own cells and getting it back. So the idea of having something tissue engineered that's off the shelf is something that people are thinking about. But it can also be achieved with iPS cells, depending on the donors. And people have done studies that if we create enough lines from enough people, then there is going to be a type of cell that's going to fit almost everybody. And so the vision of having off-the-shelf tissues that can match most people is really possible with this technology. So given that, what are people currently doing and thinking about in terms of tissue engineering? And I want to break it down into three major approaches, three major buckets of ways of thinking about building an organ that might be potentially used or therapeutic or implanted. One I'm going to call prescribed design. The other one I'm going to describe as decellularized scaffold. And at the end, I'm going to talk about the concept of self-assembly. So what do I mean by prescribed design? And I'm going to, for each of these, I'm going to talk about one recent paper that's been published in the literature on the topic. It doesn't mean that this paper is the best paper or the only paper. I just want to give an example of what the cutting edge is, what people are doing, what scientists are doing when they're thinking about these problems. So an example of prescribed design is having an idea of what you want to create and then having the technology to create that. And one of the main challenges in tissue engineering is figuring out how to get blood delivery to all the cells, especially if you're building a thick tissue. So think back to that picture of the microscopic level of the liver, where you have all these blood channels and then all these cell plates. How can we make that? How can we get blood into a thick tissue so that they get the nutrients and the oxygen? And so this is where, if you can design something, the concept of 3D printing, 3D bioprinting, if you can design something using a computer program and figure out how to make it, then you can design that and create that. And 3D printing is printing, if you think of the old style dot matrix printers, it's basically a printer jet and then it prints like a dot at a time. And then these dots of cells or dots of extracellular matrix then fuse together. And then you print the next layer on top until it kind of builds up like that. And as you can see, as you can imagine, one of the challenges right now for liver tissue for tissue engineering using that approach is that it's rather slow, and keeping cells healthy and functioning during that printing process is difficult. So what if you can print faster and you can print in sheets? And so this is where this paper comes in, where they talk about printing in sheets. It's called projection photolithography, where you can induce cross-linking of these hydrogels a sheet at a time. And this is an example of them printing something that's like a chest rook. And by projecting light and just giving the right pattern of light in layers, they can print in sheets so that they create this object. And to go from that, what they would be interested in tissue engineering is printing tubes to begin with, because we want to print complex vessels and vessels that can interact with each other and can deliver the nutrients, the complex capillaries. And they actually found that if you change these hydrogel with a photoabsorber, that means absorb some of the light so that you can control the penetrance of the light a little bit. Whereas if you don't, you can form these more intricate tubes, these smaller tubes. Whereas if you don't have the photoabsorber, the tube kind of just collapses on itself because it's so soft, and the polymerization reaction just kind of comes in. Well, using that approach, they are able to print something more complex, like a tube with valves, like valves that you could in a vein. So using that photomasking technique, they can print up through a sheet and have these valves over here, and they can test it so that from these valves will actually open with blood flow with the hydrogel material that they used. And if you flow something through it, they can actually stop the flow from going backwards, which is what we want valves to do when it comes to a vessel. They could print more complex patterns because these tubes can stay open in a smaller diameter, and they want to really test the limits of, can we press things that twist around other things and be more like capillaries or things that are close, right? So because we're thinking about the concept of gas exchange, right? And so they design another photomask where they design these channels to encircle this structure. And you can imagine that this could be a complex, a simulation of a complex capillary network, right? That they're trying to recapitulate with printing, with this precise control over this printing technology, this lithography technology. And you can go on and try to print other complex structures, like an alveolus, which is a subunit of the lung, and try to print blood vessels or things that look like blood vessels around the lung, the alveolus that does the gas exchange. Right? So if you can design it in a computer program and then have your photolithography machine print those sheets by the different photomasking structures. And this is what they can come up with. They could have this expanding sort of moving alveolus that changes with the air pressure, and they could flow this red fluid down there to symbolizes the circulation. And the concept is that eventually you just get more and more complex, right? If you can control the printing, you can print fast, then you can print something much more intricate, maybe with several units of these intertwining vasculatures and air exchange. Gas exchange units. The same report also extended this to the liver, using this technology to print a flat plate of vessels that's lined with vessels, and then the green is cells, liver cells that might be fed by the vasculature. So what are the advantages and disadvantages of this approach? Well, I think the big advantage is that you have the control of your design. You can think about what you want to create and then make what it is that you've thought up. Some of the challenges are that despite how complex and remarkable these structures are, they may still not be complex enough biologically. I mean, it's those structures that I just showed you is pretty incredible, but the lung and the liver is even more complex than that. And it is possible that the technology is, and I'm not possible, very certain that the technology is going to get better and better. And maybe at one point we're going to be able to really control at the single cell level where cells are deposited and truly print a highly complex circulatory system. But we're not quite there yet. So the other approach that I wanted to highlight is this idea of decellularized scaffold. So the idea here is that maybe it's too complex to recapitulate nature from the ground, from zero. Why don't we just start with a scaffold that's already there, that nature has provided? And so this is a study that used livers from pigs as the scaffold, as the sort of the building material to put human cells on. So the idea is from the donor animal, you have this liver, and then you can actually digest away the cells that are from the pig origin, so that then it just becomes all white. Anything that's left is just the collagen, the support proteins that make up the structure, but the cells are gone. Then you recellularize it with human cells. And in this case, they're just testing human endothelial cells. These are the cells that line blood vessels. And so then you have a reseeding process where you replace just the protein scaffold with human cells. And the advantage there is that, well, you're starting with something that nature has built, right? It has the veins, it has these big tubes that a surgeon can sew in. It has the small microvasculature that's within the liver, that's there with the complexity. And after you recellularize it with human cells, you can actually put it back. And in this case, they put it back in another pig that they gave immunosuppressive medications to. But they chose a location that is very similar to the original liver, but just slightly lower. And so this is then very much like what surgeons are used to doing for transplantation. They're similar, right? They could put in this white, that's the decellularized liver that they put new cells in. They sew in the graft, and then when they unclamp the clamps, you will see that it fills up with blood and becomes red immediately. And that's very gratifying. That's what happens in a transplant. When you unclamp those clamps, the liver starts making bile almost immediately. In this study, after they did the implantation, they gave one group, just observed the one group, and then they gave another group medications to suppress the immune system, to dampen the immune system, because we now have human cells in a pig body, in a pig organ, or in a pig animal. And they found that the grafts last a little bit longer when you give those immunosuppressive medications, but not that much longer. The longest survival was about 20 days. And still, this is in the beginning exploratory phases, but it's still pretty incredible that it can last as long as that. Right? So when we think about the approaches of using this generalized approach of tissue engineering using a decellularized scaffold, the advantages are you're starting with a normal matrix or a biological matrix. It might not be human proteins in there, but it's actually close enough that the cells are actually used to seeing this, rather than some chemical hydrogel that is not native but might be biocompatible. The other advantage is that all the complex branching patterns and microarchitecture is there. and all that complexity exists in this natural decellularized scaffold as opposed to the prescribed design approach that I previously talked about, that you have to figure out your technology of how to actually print all those things. The challenges here is that it still requires the scaffold to come from an organism, human or animal. It's not something that you build de novo. You still rely on either a human or animal source. If you use an animal source, there's concerns about cross-species immune issues and infectious barriers. And there's still challenges to how to get cells back into decellularized scaffold effectively. The third general area that I want to describe is the concept of self-assembly, meaning that we're going to give the cells a chance to signal to each other and do what they innately want to do as part of the tissue engineering strategy. And this is a paper published just a couple of years ago looking at organoids from the sort of the liver, pancreas, bile origin, and really taking a cue from development, how organs develop. And what that found is that if and this is starting from the induced pluripotent stem cells that I sort of introduced earlier in the talk. They found that if you create one kind of organoid that's, and these are sort of early on in development where the liver and pancreas and bile ducts haven't really formed but about to form. If you give them the factors to make an organoid that's a little bit more toward the anterior gut or one particular type of gut cells, and then another type of organoid, the posterior gut spheroid, which is slightly different aspect of differentiation, and you put them together, what they found is that at the interface where these two different types of cells are actually communicating to each other by some cell-to-cell or short-term diffuse signals, that this is where there's changes, where the new cell types, where the new differentiation occurs. And again, this is putting two slightly different types of steroids together. And through days, what they found is at the intersection, the cell morphologies change. They start organizing themselves differently. And all the changes is happening right at the interface where these two cells touch. And from following just what these cells are doing, signaling to each other, you get progenitors of bile ducts, you get progenitors of the pancreas, you get progenitors of the liver, you get progenitors of the pancreas. So I want to show this. This is an artist's rendition of what they have found. It's not exactly the science, what happened in their dish, but they use this to sort of illustrate their point. Right? You have the two kinds of organoids, and they come together, and something happens at the interface. And then from there, more differentiation happens. Like all these programs kick in, and the cells start doing other things, and we don't completely understand every step, but we see complexity happening. Now, the reason I want to show this video is you see how these organoids just seems to be floating, and in this space. Is that really what happened in their experiments? Well, in their experiment, this was in a matrix flat on a dish, and the cells were doing it. But in this artist rendition, the structure, and this seems to be happening sort of free floating in this space. Imagine if you were really going to put this organoid in in a culture dish. What it really does on Earth, it's going to sink to the bottom, right? So I just want to plant that seed in terms of, even this artist's imagination of what's happening seems to sort of put this, what the interaction of these organoids in a suspended space. So why does tissue engineering in space make sense? And why am I interested in that? Well, I want to show you this. And up to this point, we're talking about solving the problem of printing complex patterns, complex tissues, complex capillary networks. This is what is in a zebrafish that happens naturally in terms of forming vessels in the hindbrain. This happens in every zebrafish over and over and over again. And you can see that there's something about these cells that make them do this, and it happens every time. Here are endothelial cells in a dish. You put an organoid in a dish, you give them a little bit of growth factor, and this is what happens. They start sprouting tubes. There's something innate about these endothelial cells that they really want to form tubes. So why not give them a chance to do that? So the idea of using microgravity or space is to take away the confusing signals like a plastic dish that doesn't exist anywhere in a human body, and let the cells interact with each other and signal to each other and give them each other the correct signals. In the lab, what we have started to use, and this is work that I started with Millie, to simulate microgravity is using these rotating wall vessel bioreactors. When in function, these vessels are completely filled with fluid and they rotate around this horizontal axis. And it creates a very low shear stress, low turbulence environment. It gives cells three-dimensional freedom to self-aggregate and self-assemble. And it simulates microgravity. And compared to liver cells that are grown on a dish, in the rotating wall vessel, cell stays round, they start forming connections with each other, they start forming these organoids. And here's where I want to consider the issue of force, that the cells sense force, and that cells not only respond to biochemical signals like growth factors and cytokines, they also respond to their environment and force in different ways, different kinds of force. And in my lab, we've been interested in the way fibrosis or stiffened matrix in a diseased liver may exert force on liver cells and how that might affect their function. Depending on how stiff the surface is that a cell contacts, it actually responds differently by their cytoskeleton changes, and that affects their function and behavior. And in just comparing the way that cells look when you put them on a soft surface versus a stiff surface, the cytoskeletal organization is very different. The cell size is very different. And when you apply different kinds of force to the cells, they respond differently. And here we're showing that with increasing stiffness as a force that cells are experiencing, you actually have decreased hepatocyte function. And that is correlated or is related to decrease in the expression of important genes. And I want to point out this gene called HNF4 alpha, or hepatocyte nuclear factor 4 alpha, because it's a very important gene that controls a lot of other genes important for hepatocyte function. And this gene is going to come back a little later. All right. So when you think about what this cell is experiencing in a dish versus these cells are experiencing in the rotating wall vessel in simulated microgravity, they look so different, and the forces on them are so different. And in fact, their functions are different. So this is work I did with Millie and published together. So the hepatocytes that are in the rotating wall vessel produce more albumin, which is an important protein that hepatocytes produce, reflective of function. And if you do a gene analysis, this is global gene analysis of what the type of genes that it's expressed in a 3D aggregate versus a monolayer. There are many, but the processes that are turned on are different from liver development and to liver regeneration. What we found is different that's upregulated in these aggregates that are formed in microgravity is a lot of functional genes. So function is increased or maintained when cells are allowed to associate with each other and form these connections with each other the way that their innate programs want them to. And when we look at what is controlling all of these genes, we get the same hit as what I showed you before, HNF or alpha. In two different systems, we have found that this one gene may be key to controlling how the hepatocyte senses force, whether it be a stiff surface or dimensionality, 2D versus 3D. This master transcriptional regulator is being affected, and that might affect gene function. So there are limitations to the rotating wall vessel or these microgravity analogs, and that's because as the particles or the organoids get bigger, you have to rotate the rotating wall vessel faster to keep them in suspension. And eventually you're not going to be able to do that because the shear stress is going to increase. And that's the thing that we want to avoid, to keep them in this idealized, suspended environment. So the size of what can be created using ground analogs of microgravity are limited. And this is a video downlink from Colombia before the last flight of the rotating wall vessel in space. And what you can see there is that the organoids that are formed in space are much larger compared to what is formed on the ground. So these are side-by-side comparisons of that experiment with photos showing that the organoids or the cell clusters that are formed on the ground are much smaller and scattered, whereas you get larger aggregates in space. And so these samples, along with the crew, was lost upon re-entry of Colombia. And all we have of this experiment are these images in that video. But there is evidence that cells behave differently when in association with each other in orbit, in low Earth orbit. My lab right now has a funded project through the NSF and CASES to investigate liver tissue engineering in space with this concept that organoids in this high-quality, sustained environment of the International Space Station will be able to form larger, more complex structures and have a window of understanding whether we can, and by taking away confusing signals, non-physiological signals, that we can form complex vascularized tissue structures. And we have a patented bioreactor design that we think will optimize cellular interactions in the space station. So self-assembly. So I put tissue engineering space in the bucket of self-assembly. And I think the pros there is that it really gives the cells a chance to follow their innate programs, and it has the potential to really emulate the biological complexity and structure that is necessary to have full function of a complex cell like the hepatocyte. And the challenges there is that it requires a deep understanding of biology. And of course, there's the question of scale, whether we can really build a large, thick structure with this approach. So I hope I convinced you that there is an unmet need in tissue engineering in healthcare, and it'd be a major change if we can figure out a way to create tissues, functional tissues outside the body that can be used to treat people. There remains barriers to create thick vascularized tissues, and that having a sustained high-quality microgravity environment may be a key tool to facilitating and facilitating tissue engineering and creating these vascularized tissues. I would like to thank the members of my lab who have been involved in the work leading up to this point, and our funding sources. And I'd like to especially acknowledge the NSF, CASIS, and NASA for their support of the specific work in tissue engineering space. All right, I'd like to take on any questions that people may have.
Thank you. That was absolutely fascinating. My co-chair, Dr. Robinson, is on as well. I'm sure he's a little nervous, thinking he probably quit flying to space just in time before his liver outgrew his brain. I'm not sure if he's going to accept that joke very well. He is going to share with me the Q&A part of this. We're looking for questions online, so audience, please don't be shy. This is a rare opportunity to speak with one of the world's experts in tissue engineering and one of the very few who've actually been able to take advantage of the microgravity environment in the ISS to prove out some of her hypotheses. I could start with one question, Tammy. This is not so far-fetched in terms, obviously this will have great transfer back to Earth, and that was the intent of your original work, is how do we improve terrestrial healthcare in this condition. But also, in the long missions that I presented last week, the deep space missions, there are considerations of what will we do on 3-year missions if we had some sort of injury with large tissue defects, burns or trauma, et cetera. Even a myocardial infarction where a significant portion of the heart muscle gets lost. Is it conceivable, way far out there, that those might be applications on Earth and in space, that there are many other tissues that might behave in ways that you've demonstrated here with liver tissue?
Absolutely, absolutely. And I think tissue engineering for tissues like skin is probably is going to happen first, even now much more advanced than something like the liver, where it might be sort of thinner sheets that is required for coverage. And I think those things, so tissues like that can, I think, more potentially be sort of a first goals of how of these applications. And certainly, it's difficult to think about what astronauts will need on a 3-year mission, and what we could provide for them. But certainly, any approach that could repair and regenerate would be very useful if we could really package it, miniaturize it, make it easy I think that would be very useful on that lonely mission.
I'm going to ask Dr. Robinson if I'm sure he has a few questions for you.
It seems to me that astronauts will be the last person to benefit from this work, actually, given how useful and exciting and relevant it is to the rest of the human race population to be able to do these large organ tissue growth. So I'm excited about it. And certainly when you think of long duration missions, you need to, it's all about preparedness. And you know the state of your vehicle and your tools and your learning. You don't know so much about the state of your body as you progress. And there's certainly challenges, and it's a hazardous environment in many ways. And you would like to know how is your body doing, and what are the options in case something serious happens.
And probably a question for both of you is, do you imagine there might be other analogs being developed on Earth? ISS is fantastic, and it's going to remain a wonderful resource to us, I think, for a long time. But Tammy, do you have other ways of creating a microgravity environment besides the small centrifuge rotating wall chamber that you showed us? And Steve, I know you've been thinking about this for a long time, because creating artificial gravity is one of the countermeasures that's talked about. So could both of you take a shot at that?
Sure. So there are different analogs, other analogs for microgravity. The one that I showed is called the rotating wall vessel, which just rotates around a horizontal axis. There's another analog that we do have in the lab, and it was a gift from Millie, called a random positioning machine. And what it does is it's a platform where you could put your tissue culture or any kind of culture dish on. And there is a computer algorithm to rotate this platform randomly in space. Wow. So that the gravity vector in the end cancels out to zero. Wow. And the advantage of using the RPM is that you can have a computer algorithm to simulate zero gravity, but you can also have a computer algorithm to simulate moon gravity, Mars gravity, and any other partial gravity in between. And so one of the projects that we're looking at is the effects of partial gravity on cell aggregation and also liver function. Because in space, it may not be an all or none, right? And in thinking about artificial gravity, I think one of the key questions is how much gravity is enough, right? How much gravity is enough to mitigate bone loss or immune dysregulation? Maybe we don't need 1G. Maybe when you get to Mars, that amount of gravity is enough, and maybe you can bounce back. Or when you're thinking about designing a spaceship, having to design a spaceship that simulates 0.5G versus 1G saves you a lot of energy to do other things, I would imagine. So I think sort of figuring out what is there a threshold of gravity that keeps biology functioning, and where that threshold is important, and it's interesting.
Yeah, it's kind of amazing what we don't know. We know a lot about 1G. We know quite a bit about, almost 0G. But for the things that happen from change in the human physiology, from the cell level up to, bone structure, for the things that change when you go from 1 to 0, what happens if you go from 1 to, 0.3 or 0.6? It's amazing how little we know.
I was also thinking about the application to food, because one of the problems on the long missions is going to be, how do we maintain adequate nutrition? And I'm wondering, Tammy, I know this is a little bit off topic from liver, but it seems to me that food sources, plants, for instance, would grow differently in microgravity. And I'm sure you've probably thought of that, but Could you speak to that for a second?
Sure, absolutely. There's a whole area of research precisely on that, uh, food production and how plants grow, um, and very much so affected by gravity. The direction of roots and stems and stuff like that. But yeah, it's uh, I think that's sort of that's the important, interesting thing about gravity in our environment is that all life evolved on Earth on 1G. And like any science, we don't know what its effect is until we take it away.
Mm-hmm.
So it's removing that variable. And up until recently, we haven't been able to investigate that very basic biology is sort of the effect of gravity. We just assume this is the way it is. This is the way life is. But um, it is the way it is because this is all we've known for hundreds of years, and we haven't had a chance to investigate. So take the variable away, the gravity variable away, and see what biology is there when it's gone. So I think that's the exciting opportunity that we're able to pursue with the colonization of low Earth orbit. that we have an opportunity to ask these questions.
So Dr. Chang, I have a question for you, kind of an operational one. Can you imagine pursuing this research using CubeSats?
Absolutely, and thank you for that question. Thank you for that question, because it is a question of scale, right? So when I was sort of discussing this idea with one of my colleagues, she said, "Well, yeah, this is interesting, but you're not really gonna build organs in space. I mean, that's not really gonna happen." And I think the implication is, well, okay, we could do these experiments, but then we have to figure out how way to simulate gravity on Earth, or simulate microgravity better on Earth, or some other approach. But as you pointed out, the economics of launch is changing. The accessibility of low Earth orbit is changing. Yes, maybe we could figure out either building a giant RPM or figuring out how to make magnetic levitation really work on Earth to create these suspended environments that's idealized on Earth. But if through these experiments we can we show that microgravity, or this way of suspending cells, is a key tool to allow them to interact with each other in their most natural state, then the economics or the ability and the technology to send things like CubeSats or sort of completely autonomous biosatellites that can utilize the low Earth orbit environment and then return safely to some location on Earth, I think it's not it's within I think within reach. Yeah. But the first is we have to show that it actually makes a difference. That is the key ingredient to making these complex vascular tissues. And then I think if we show that, I think there's actually a lot of interest in manufacturing in space, because there are certain advantages. And if we show that microgravity is a key tool for tissue engineering, and that it is the most efficient and potentially economical way of generating these large-scale tissues, I think that there will be interest in pursuing that.
And Tammy, just in the sequence of things, would your next step to take some of the tissue and then implant it to, try to put it into the context of a really larger scale biological functioning system?
Absolutely. Our proposed project NSF cases, the liver tissue engineering project actually has a component where we're proposing to bring back the vascularized tissue, or the tissue that we generate in space, back live, and be able to implant into rats as a test of function. Of course, we have a lot of backup plans in case we can't bring back live tissue. But that's one of our goals, is actually to be able to implant it and test function.
And have you had success doing something like this deep underwater? So sort of suspended, not truly microgravity, but I'm sure a lot of people are thinking, well, it might be easier to get something in the ocean than up to the ISS. Could you talk about that?
That's actually an interesting idea. I actually never actually have thought of that, that approach. But I think you bring up an important point, which is the idea of how buoyancy as a force is maybe simulating microgravity. And that is actually something, a thought that we're taking some inspiration from. Because when you think about, buoyancy, we're training in water, is how astronauts train to get used to a feeling of microgravity, right? So it's different, but some aspects of it are similar. And so when you take it sort of down to a cell, when you think about development, because life on Earth evolved in an aqueous environment in water, whether you're a fish egg in the water, or you're a mammal and you're a single cell in a mother's womb, you're in an aqueous environment as a single cell. And there's the buoyancy force on that. And as the single cell becomes 2 cells and 4 cells and 8 cells and 16 cells and up to a fetus, the buoyancy force on that and the fluid around that changes. And how is that force being perceived by the developing embryo? How is that force being perceived and regulating organogenesis? I think that is something that we're drawing some inspiration from, and also what we're sort of thinking about when we're thinking about using microgravity as a tool for tissue engineering. So if you want to think about us trying to recapitulate or simulate what cells might be signaling to each other during organogenesis, well, maybe in the beginning it has to be very, very buoyant or very low gravity, right? But then as you get to a larger and larger structure and those things are being formed, maybe you want to start adding back that gravity, or decrease that buoyancy force, because maybe those are the signals that's going to mature and keep things functioning, right? So these are ideas, these are conjectures, but certainly I think there could be a connection in terms of buoyancy and microgravity, and I think, as we show, sort of all of this has to do with how the cell perceives its environment and how it perceives force, and the force can come in many ways. As I've shown, is sort of in how stiff a surface is, or if it's touching other cells versus in a 3-dimensional environment versus a 2-dimensional environment. But what it might come down to might be the same signaling mechanisms, right? And in terms of regulating function. So
what's fascinating to think of the ISS as a uterus. Maybe that's why they refer to it as the mothership sometimes. Steve, do you see questions in the Q&A? Because I'm not able to open it, but if you see one, oh, you're nodding. All right, take it away.
Yes, from Dr. Sonia Patel. What impact does radiation in space have on these cells and the experiments?
Oh, I think that's a great question, and a question that I cannot answer. But I think there's an active investigation on the effect of radiation. As you may be alluding to, radiation and radiation damage is a key risk, an important risk for long-term space travel, what it can do, not only in its potential to cause downstream carcinogenesis, but it can cause more immediate degenerative problems, including fibrosis and other tissue damage. And there is some evidence that prolonged radiation, specifically to the liver, make like any other tumorigenesis or carcinogenesis, may increase risk of liver cancer or liver tumors down the line, but it can also, because of the inflammatory damage, cause fibrosis, which is the final common pathway to a liver failing. And I think those same risks and concerns are with every part of every organ in the body. And I think it's a very active area and a focus of research for NASA, who's thinking about sending humans into deep space.
And Tammy, the idea about looking at other types of organisms is so fascinating to me, that whole field of biomimicry. And I sometimes think of the liver a little bit like a sponge, and I think about the sponge and how they grow in so many different forms and shapes underwater. Are there models, biologic models, that you think of in a biomimicry sense that represent some of the work that you're doing?
What we are modeling sort of on the small level is using the organoids. And when we use the organoids, we don't only add in liver cells, which is one cell type, but we also add in the endothelial cells, which is the cell type from blood vessels. And we add in another cell type called the mesenchymal stem cell, which is a stem cell that gives rise to things like fibroblasts, or these stromal cells that produce the matrix. And what we're finding is that all three cell types are important in creating the organoid and supporting function. What we're actively investigating and trying to figure out more in detail is what cell is doing what, and exactly what roles they play in supporting each other.
Well, I think it's absolutely fascinating, the idea that not necessarily, the goal isn't necessarily to create a whole organ, but if you even can create functioning tissue and you could percutaneously inject it into defects in organs, that concept is absolutely fascinating and relevant. We're doing it with other types of things, but to actually have the functioning biologic tissue that belongs there and inject that in is really remarkable.
Absolutely, and I think that's a concurrent or parallel approach that is being investigated. So there's the idea of creating something outside of the body, then we could put into the body. But then a parallel approach that our lab is also investigating is getting the body to do it itself. And kind of exactly what you're suggesting is creating the environment, or creating the room or the niche for engraftment of stem cell derived cells.
So the body's the bioreactor.
Yeah, absolutely.
Steve, you have another question? See your mic coming off.
No, there's no more questions in the chat here. I was really touched to see your photographs at the beginning of the lecture, Dr. Chang. I was on the mission after Columbia, the first one, and my final mission in space was 130, the one right before you, the one you saw.
Yeah. Wow. those 2 weeks at Kennedy, I think, is one of the most memorable experiences in my life, and yeah, that's you know, Millie not only gave me the opportunity to sort of really launched my career in this field, but also gave me the opportunity for once in a lifetime opportunities. It experiences and going to the Kennedy Space Center, being able to go in there and just flash my badge and be let in every day. I felt so proud, you know, just being in that
Yeah.
environment and doing that. And it was a wonderful experience.
I agree. You know, I had a NASA badge for 37 years, and you know, you would think it was the hardware and the rockets and the spacecraft and stuff that's the exciting part, but really it's the intensity and the audacity of the human intellect and spirit that always got me excited.
Wow. There is one more question I'm going to slide in here, Tammy, because it's such a rare opportunity to be able to talk with you and also have Steve here. The question is, are you able to take any lessons from living donor liver transplantation in liver tissue engineering?
Oh, absolutely. Lessons all the time. And I think that's why it's an evolution. I think we needed to know how to do transplants before we do liver tissue engineering. I think that's where all the lessons are. Because we know how to put another human liver in another human body, that is where we're taking off from when we do tissue engineering. Absolutely.
And I think as we close this out, I want the audience, and I certainly appreciate the fact that you are one of the people I define as the intersection people, where these major inflection points happen because you have a foot in the clinical world. You do liver transplants. You're on the front lines of that, as well as your research hat and your innovation hat. And it really does create an acceleration or an inflection point and advances the field. So I thank you. Steve, thank you so much for co-hosting this with me. And Tammy, thank you for sharing the personal side of your career and your journey. And again, our big thanks to Millie for advancing your career, seeing your potential, and facilitating this. And as I say over and over, she's created a legacy of generations of scientists, and particularly women scientists and surgeons and space explorers, that's going to impact going forward many generations. And she's handed the baton to you. So thank you for sharing your time with us tonight. Thanks to everyone in the audience, because these evening sessions are precious time away from other things you'd be doing. But thank you for your curiosity and your questions and your attention. And we'll be back next week.
Thanks, Dr. Chang, and thanks everybody for coming to class.
Thank you for the opportunity. Thank you.